All right, I think that's enough time to have and let everybody get kind of settled. So with that being said, I want to welcome all of our fantastic guests that are watching from home or wherever you might be. My name is Brett McAllister. I'm the Director for Graduate Admissions for the wonderful Halmos College of Arts and Sciences. With me this evening from my staff is Miss Megan Troy, who a lot of you will know from any conversations with admissions office about your application or any questions about the program to get you started. With us also is a surprised guest in Dr. Reza, who is the department chair for our fantastic, new-ish titled Department of Chemistry and Physics and Engineering. Engineering recently moved over from undergrad standpoint. Now, all three of us are very excited because I get to introduce our fantastic guest lecturer this evening, Dr. Tara Ghafourian. So Dr. Ghafourian is an associate professor at the Department of Pharmaceutical Sciences, the B&J College of Pharmacy at NSU. She specializes in drug discovery and drug delivery using molecular modeling, machine learning, and in vitro assessments, as well as formulation development. She obtained her Ph.D. in pharmaceutical sciences from Liverpool John Moores University in the United Kingdom and is awarded a postdoctoral fellowship to estimate the endocrine disruption of environmental pollutants. Now, before joining NSU in 2022, she held academic positions in the United Kingdom since 2001, including the University of Kent for 11 years, University of Sussex for 5, and University of Bedfordshire for 2 years. Dr. Ghafourian is also the associate editor of Springer's Molecular Diversity and an editorial board member of several peer-reviewed journals. Most recently, her research has focused on the application of machine learning to the discovery of biomarkers for Alzheimer's disease, prediction of mental health-related drug effects and side effects, and modeling of mitochondrial function. Now, her title for today and the topic of her lecture will be Success and Challenges of Artificial Intelligence and Drug Discovery. So, Doctor, thank you very, very much for being here with us this evening. I'm excited to learn, and our students are ready to as well. So with that, we will have a 10 to 15-minute Q&A at the end, and I'm looking forward to learning more about this topic. Doctor, the floor is yours.
Hey, thank you very much for inviting me, and thank you very much also for presenting. I have put my first slide. Sorry. So my first slide is a brief biography, but because you already explained my biography, I will skip this one and move on to the topic of today's discussion or today's lecture. So all of us by now have heard about artificial intelligence, and we know the importance of artificial intelligence in today's economic. So for the first introductory slide, I have picked some of most recent news about artificial intelligence development that everyone is aware of. But the outline of my lecture otherwise is going to include introduction, and then it will be followed by, my introduction is going to be in 2 sections. First, introduction to drug discovery itself, the process of drug discovery. And then I'm going to explain about scientific modeling and where AI fits within the scientific modeling, because AI is a subtype of scientific modeling. I'm going to give some basic examples of computer-based drug discovery, which is data science related like AI. And then I'm going to move on into current progress made in computational modeling and the reasons for those, which includes databases, availability of current databases, widely availability and larger databases that are available, examples of advanced computational modeling, and then I will move on to some case studies and examples of success and challenges. And then I have a concluding slide to summarize all of those. I will start with FDA Modernization Act, which was signed as part of an omnibus bill in 2022 by President Biden. And basically, the FDA Modernization Act eliminates the requirement for the conduct of animal testing before human trials. So basically, drug discovery process always involves animal testing before FDA can allow any human clinical trials. But due to recent progress in not only artificial intelligence, but also other technology, which I will mention in the next slide and some others as well. This Modernization Act states that animal experimentation is no longer necessary if the companies, the pharmaceutical companies, can provide enough evidence to show that the drug will be safe when tested in humans by other methods. I have picked some quotations from FDA Commissioner. He said that for too long, drug manufacturers have performed additional animal testing of drugs, even when it wasn't necessary because of because we already had data in broad human use internationally. So it could be data because the drug has been used for other diseases, or drug has been used in other countries, other geographical locations. So sometimes we already have human data. And despite that, before pharmaceutical companies had to do animal testing, and it's no longer required by FDA. So FDA has given incentives to the use of advanced computer simulations to predict a drug's behavior, along with human-based lab models. For example, we have organ on a chip systems where we have a model, basically a physical model in a chip of various human organs that you can test the drug and see the impact in terms of the toxicity or even efficacy of the drugs on these cells taken, different cells from the same organ or cells from various different organs linked together on this single chip. We have three-dimensional cell cultures, and we have high throughput screening data from these systems that are available and can be used in using advanced computer simulation, can be used for modeling and generating artificial intelligence systems that will then predict whether a drug is safe or not to be tested on humans during the first phase of clinical trials. So this picture here shows, for example, a sequence of amino acids that could be part of an antibody or an antibody antibody intended as a drug, a biological drug. So based on the sequence of amino acids, we should be able to predict its surface properties and its three-dimensional structure of the folding of this amino acid. Therefore, a lot of properties like aggregation and solubility and viscosity and also we can predict based on the surface property, we can predict which body organs in human it will distribute to. And therefore, we can predict based on these distribution pattern and also surface properties and the molecular composition, we should be able to predict how safe or how harmful the drug could be. Will it bind to certain proteins in human body or not? Will it target just certain intended proteins, which are the intended target of these antibodies or not? So this is just one example that could be found in FDA's announcement. Right. In terms of the introduction to drug discovery, I think this slide is necessary because for those of you who are not familiar with the drug discovery process. So currently, drug discovery process starts with an unmet medical need. So first of all, we have a disease and we don't know how to cure it, and we are working on it. We want to identify what proteins are involved in the course of the disease. So we may identify several proteins that are upregulated or downregulated during the disease. And then we may decide to target these proteins, but we need to then first identify whether it is safe to target these proteins. And that is an important part of drug discovery, and it is called target discovery, which is currently being improved a lot, because a lot of in other areas their progress was already there. So I will get back to this in the later slides, but the use of computer-aided drug discovery has been there for years and for ages. However, application to biological systems have been less advanced due to lack of enough data, which is becoming more common these days, which is becoming more available these days. I mean, the data becoming more available these days. So basically, we have an unmet medical need, and then we need to discover proteins that are involved in the course of the disease, and we then identify within these proteins suitable targets that are safe to be targeted. It will not harm the patient if we reduce the amount of these proteins, if we inhibit these proteins, etc. Next is, after identification of a target, is to discover agents, chemicals, hopefully small molecule chemicals, because they are cheaper and they are more flexible. We can change them, we can manipulate them, and they are cheap. So we want to find some small molecules that are able to target these proteins and bind to these targets, and hopefully inhibit or potentiate their actions. Normally inhibit, because inhibitors are more sought after than potentiators of proteins actions. So once we identify these drugs or drug candidates, we will test them to find whether they are safe or not. And the testing normally starts with cheaper testing protocols, for example, cell-based methods, or methods, any in vitro method that doesn't require a whole animal. So normally we start with these simple, cheap tests, and then we move on to animal studies, which is the next level, because they take longer duration and they are more expensive. And also, we don't want to use animals just for any chemical. We want to make sure that from other studies that these drugs are probably useful, probably efficacious, and safe enough, and then test it on animals, which are more expensive investigations, require more resources, both in terms of the availability of animals, the length of studies, and also based on the expertise required to do this. And then we modify the drug structures if they are not safe, to optimize their properties, add groups, chemical groups. We may add a OH group, or we may add a methyl group to make it better absorbed, less toxic to the liver, and so on. So we optimize these drug structures, and then once they are optimized and have been proven and has been proven, because we normally pick one that is the best one, and then we move on to the next level, next stage, which is formulation and scale up. And then at that stage, we can file IND, investigative new drug application, to FDA. So we apply to FDA and say that, okay, I have this drug, I have run all these tests, and it will be quite a lot of these test results that will be submitted to FDA, so that FDA can find the drug safe to be tested in humans. And then the first in-human clinical trials will start by recruiting patients, and these clinical trials are very expensive, as the one clinical trial could be as expensive as everything else that has been done before this stage, all the scientific investigations and so on. After clinical trials are successful, the 3 phases of clinical trials, the company will file new drug application to FDA, asking for approval, regulatory approval for marketing. And then once in the market, the company will still follow the drug, will do post-marketing surveillance to see if there are any side effects that they didn't detect during clinical trial or not. All these, So all these steps that I just mentioned, the ones that I have highlighted with an arrow, are stages of drug discovery and development that can be helped and expedited by the use of artificial intelligence. And I'm going to give some examples later. But what is also to note and noteworthy is that all of these experimentations are modeling, because we are using a type of model. So basically, we either use in vitro models, if we have a receptor on these cells and we are running cell-based binding investigation, which is the case that I have shown in this picture in the left-hand side. This is an in vitro model of what will happen in real patient population, right? The next one, we have animal models. Animal models are not real, obviously. They are modeling and hopefully modeling to the best they can to predict what will happen in human. And we also need to remember that all of these models have errors. Animal models concordance with human on average is around 50%. There is only 50% concordance between animal studies and human study results. But we do them because we still need these models to be able to predict. Next, we have clinical trials. Clinical trials are still models. They are still models because they are just representative of the population, but they are not real patient populations. They have been handpicked to have, to have certain age range, a control group and the patient group, or the placebo group and the patient and the real drug group will need to have a similar age range. They will obviously they pick them so that they are not too old or too young. They don't pick pregnant people. They don't pick people who may have other diseases, other concomitant diseases. They will not choose if the patients are taking other medications. So they are handpicked individuals for comparison reason, and they are not the true representative of the whole society, because the whole society will be using the drug in different ways. They might forget to use them. They have different age groups. They might use it for longer duration, etc. And then we have computational models. So you see, all of these are modeling techniques, and as long as computational models can prove that they have similar levels of accuracy to other methods of modeling, experimental modeling, they should be allowed to be used. I have here a quotation from a scientist that I admire a lot, a polymath. Von Neumann states that all we do in science is modeling. So basically, according to him, scientists, they mainly make models. That is the job of a scientist. That's what we do in science. By model, we mean a mathematical construct, and with the help of some text to interpret and to describe this mathematical construct. And the justification for this model is that it works. Okay, and by it works, I mean it has good level of accuracy. So the justification of such a mathematical construct is solely and precisely that it is expected to work, that is, correctly to describe the phenomena. So that is all we do in science anyway. Now the introduction to artificial intelligence. In artificial intelligence, we have the definition is that they are computer systems that can perform tasks that typically require human intelligence. For example, image recognition that you see in the right-hand side, some of the examples, image recognition, speech recognition, decision-making, natural language processing, text generation, such as ChatGPT, that everyone is familiar with. translation of text from different languages to others, video, sound, and image generation. So these are examples of artificial intelligence. Then we have generative AI. Generative AI not only makes predictions, they can also predict, they can also generate new data. They can generate text, they can generate images, they can generate music, or even videos these days. And I have given examples of some of the systems in the text here. Then there is something called or terminology foundation models. Foundation models, by definition, based on the US new executive order, is AI models trained on broad data. So in the next slide, you will see that we have various levels of artificial intelligence. So these are the ones that are developed and trained on broad data, all encompassing different types of data. They use self-supervision, contain at least tens of billions of parameters. So they are very, very complicated, are applicable across a wide range of contexts. So ChatGPT would be an example of this. Then you may have heard about ML or machine learning. What is the relation between machine learning and AI? So these days, some people use them interchangeably. However, they are slightly different in that ML or machine learning specifically refers to the algorithms that are used to apply to the data, to train the data, and to make the predictions. So the algorithms are called machine learning, whereas AI includes the data and the whole system and the computer system. You see here where it fits within this artificial intelligence system in the picture. We have a database of images, and then these images are deciphered in terms of generating features. We can identify certain points in these images that are useful in recognizing and classifying cats and dogs. So this is the first step that is done during this artificial intelligence system generation. And the next step is that these features, these data points, or these points in the image that have been identified, they will be used in a machine learning algorithm to identify whether the object, or to classify whether the object is a cat or a dog, or in general, what the object is. So this is the normal artificial intelligence and where machine learning fits. However, we have a new, more recent concept or more recent method, in fact, which is called deep learning. Deep learning is a subtype of machine learning, but it is more powerful and more complicated in that it doesn't require that we first generate features. So images will directly be used in these systems. Normally, they are convolutional neural networks systems. They will automatically identify and learn features, and then run algorithms to predict whether the image is what type of object the image is. And as I mentioned in the previous slide, we have different levels of artificial intelligence. We have specific artificial intelligence, which are smaller systems that are specific to the task. For example, I may want to just predict whether a drug can inhibit certain enzyme, dihydrofolate reductase enzyme. I want to predict whether the list of drugs that I have, the list of molecules, not drugs even. So the list of molecules that I'm going to synthesize and haven't synthesized yet, I want to predict whether they will be able to inhibit dihydrofolate reductase enzyme. So I can use this AI system that's specifically designed for this task. This will be called specific AI, and that is most prevalent and most widely used in science and even in drug discovery today. And then we have universal AI, which is intelligence across a variety of tasks, and that will include ChatGPT. And then we have superhuman AI, which is hypothetical and a topic of debate because we don't know if it is possible or not, for sure. ML systems can be divided into different groups as well. We have supervised learning, and we have unsupervised learning, and we have reinforcement learning. And I think it is too detailed, and we don't have time for me to go and explain all of those. Machine learning, as you see in this picture, hypothetically, machine learning systems or algorithms discover patterns in data, and then they can use the way or the process to apply to future instances and predict the shape of the future objects. So they are systems that have been designed for pattern recognition. So they learn from the data that's available to them, and then they predict the future. So they don't invent anything new. They are just apply the learned knowledge to future instances. Okay, I need also to mention that in drug discovery, we don't just use data modeling. So far, artificial intelligence is about data and about fitting a model to the data, fitting an algorithm to the data. And it is also called data modeling. But not all modeling in science and in drug discovery is data modeling. We have simulation types of models, which are quite commonly used and perhaps more commonly used up to now for scientific purposes. The simulation models start with a hypothesis. So we already have a hypothesis. We don't need to have a lot of data because we know theoretically that it is correct. For example, rules of physics are like that. In this picture, in the right-hand side, you see the distance moved per minute as a function of the car speed. So even if you don't have a lot of data, you know that such a relation exists. And it is a physical relationship, based on the physics, based on the hypothesis. And then we know there is a causality because we know the mechanisms. However, in data modeling, we don't have any hypothesis. That's why it is so important that we validate these models. For example, we may collect data about consumption of chicken in a country or in a place, in a neighborhood or in a shop, okay? And then we also collect data on the amount of imported oil in barrels, okay? Imported oil. And then we collect the data over a certain period of time, say data for each year, and then we can put these in a scattered plot to see if there is a relationship between them by any chance, okay? And then we have a scattered plot, then we fit the line of best fit and see, oh, there is a trend that's obvious here. So there is probably a relation, a correlation between these two. However, this doesn't state that there is a causation. This is just a correlation unless you have other methods of verifying that this is a correct, first of all, a correct correlation and can be used for future as well. In the future, if you know the amount of imported oil, can you predict then how much chicken is going to be consumed or not? And also, you need to validate in terms of the causality. Do you have any reason to believe that this is the cause of the amount of chicken consumption or not? So there are advantages and disadvantages to data modeling, and AI is a type of data modeling, just a very complicated method of data modeling. As applied to drug discovery, data modeling or artificial intelligence will require a lot of data. So we have data for chemicals, we have data for biomedical, in terms of biomedical data of these chemicals. So first, we may have molecular weights, we may have lipophilicity of these chemicals, we may have length of the molecules or the size, surface area, number of oxygen atoms, number of polar groups, number of hydrogen bonding groups, etc. So we may have, and it is easily generatable by computer software. So we have chemical data and physical chemical data, and also we have biomedical data on these drugs as well. We may identify which enzymes they bind to, or what is the potency of these when inhibiting certain enzymes or certain receptors, etc. So we have biomedical data as well. And then we have types of data which have become common these days as well. Electronic health records or other types of clinical data, which are becoming more and more available as well. These types of data then can be used for machine learning, because when you look at the data, you will not, a human eye cannot detect any patterns, because we have so huge volumes of data, as well as it is whatever we do will not be quantitative. So we need a machine learning algorithm to apply to these to then identify patterns that are otherwise difficult to identify. These patterns then can lead to predictions and decisions for new questions. And also these patterns in themselves are knowledge, because we then use these patterns to discover better agents. For example, a pattern may emerge that if molecules are flat, they may bind better to certain enzyme. Then this is a knowledge that we can use in future drug discovery and make our drugs more flat, for example. In terms of AI and computational modeling history, we know earliest computer, which was a mechanical machine to do calculations, started in 1822. So it's been around for long time. Then in around 1950, we had a movement, more attention to data due to World War II. We had Alan Turing, who worked on decoding Nazi Germany codes and deciphering their messages. And he basically produced this mechanically working computer and artificial intelligence, the first basic artificial intelligence. Later on, this progressed further, and we had algorithms and machine learning, and the type of artificial intelligence which was called symbolic AI, which is a basic type of AI that is based on logic. So logic-based AI, which developed further until 1990s, but then it couldn't go further. New methods and new architectures emerged since, before, during these years that took over. So these were the basis of expert systems that you may have come across in, equipment for basic decision making. Around the same time, in terms of drug discovery, we had a scientist called Corwin Hunch, which a California-based scientist, and he first published a paper and coined the terminology quantitative structure activity relationships, or QSAR for short. So quantitative structure activity relationships is the basic form of data science applied to drug discovery. So it is a data analysis based modeling system. I will explain this with an example, with a very basic example from Corwin Hunch in the next slide. So you see, data science has been applied to drug discovery since then, although under a different terminology, and also using limited data and also simpler methods. Since 2000, the year 2000, we had a huge progress made in machine learning methods and especially with regards to artificial neural networks. Until then later on, we had foundation of generative AI, and then we had AlphaFold, which I will explain, prediction of protein folding in 2020, and ChatGPT, which was launched in 2022. So the basic form of AI, if we can call it that, was a QSAR method developed by Corwin Hunch. This one is a model that was published in 1968. For the first time, Corwin Hunch applied several different X's or in a multiple regression analysis. So fitting a line of best fit to data is quite common, or has been always very common in science. But Corwin Hunch, for the first time, suggested multiple regression analysis for this type of problem. In this example, adrenergic blocking activity of a group of chemical beta haloalkylamines, which you see in the picture as well here. The adrenergic blocking activity has been measured for a group of 22 of these chemicals. These chemicals have different X groups and Y groups. And then based on the lipophilicity of these groups, of the attached substituents, attached groups, and electron directing property, which is measured by sigma. So lipophilicity is measured by pi, and electron directing property is measured by sigma. Then he was able to predict and draw a line of best fit. And then based on this equation that he obtained from the line of best fit, we could say that, or he could explain that, if we put more lipophilic substituents, if we put lipophilic substituents on X and Y, the drugs will be more effective as inhibitors. And if we put more electron donating groups, again, these chemicals will be more effective adrenergic blocking agents. And this is one of the earliest examples. But since then, so back then they had to actually measure lipophilicity and measure the electron directing effects using laboratory experimentations. But since then, computer simulations and computer modeling has progressed a lot. So a lot of properties can be calculated, chemical properties can be calculated using computer software. And by then predicting these molecular properties, we then use them in algorithms, using machine learning algorithms to predict the toxicological properties of these chemicals, the pharmacological properties, absorption and solubility, and absorption from skin, and also pharmaceutical processes, whether they will be the drugs will be released fast from polymer matrix in tablets or more slowly, so the rate of release can be predicted, and so on. So, and this can be done because there is a relationship between molecular structure and properties, because molecular structure determines whether the molecule is going to be soluble or not, whether the molecule is hydrophobic or not, and whether the molecule will bind to this enzyme or not. Shape and lipophilicity and hydrophilicity and presence of polar groups, and the three-dimensional shape determines all these bindings in human body. An example, a more recent example of a QSAR work in my lab, done by my PhD student, published in 2020, is prediction of inhibitors of an enzyme which is called alternative oxidase, or AOX for short, as antifungals. So the fungus contains an enzyme called alternative oxidase. It is not present in human. In human, there are other forms of oxidase enzymes, but not this alternative oxidase. And my student measured first if these drugs are inhibiting alternative oxidase or not. So she tested around 50 chemicals against alternative oxidase enzyme. Using this equipment here, I'm not going to go into details. So basically it measures respiration or oxygen consumption rate. And then she generated computer models of these molecules in the computer and calculated molecular properties of these molecules. And then based on molecular properties, she was then able to generate a QSAR equation, which basically says what groups or what form of the structure can have better inhibitory potential. So for example, here you see neutral form, because this is the easiest to explain. So you see that it has a negative coefficient, and it basically means that if neutral form is high, these molecules will be less potent. Therefore, because for binding, they need to be acidic. So the more acidic they are, the better the binding. And with that, we can then, this is a knowledge that we have gained based on this equation, we can then move on and develop better and more potent inhibitors based on this knowledge. She also did other ways of modeling. She measured binding of these individual chemicals one by one against alternative oxidase enzyme, the three-dimensional model of alternative oxidase, by using molecular docking software. And these software are available. Some of them are freely available. Some of them are commercial. However, these docking investigations are also useful, because then she was able to identify which amino acid residues in the binding site or the active site of the enzyme needs to be attached to the drug molecule so that the binding is effective and inhibitory. So by the use of these two methods, we were able to predict future inhibitors of alternative oxidase. So you see, these are models, and you could call them still very, very basic artificial intelligence models. The reason why I call them basic is because they are simple correlations. They are not sophisticated systems. However, if you ask computer scientists, they will tell you that we always look at the size of the data, then fit a model. We will not be able to fit, I mean, we can fit complicated models to these small data sets, databases. But the prediction, future predictions might not be good, because the more complicated the model is, the more chance of, or the higher the risk of chance correlations, chance relations that are not valid and they are not real. But progress has been made in terms of abundance of template three-dimensional structures in proteins, growth of virtual compound libraries, and they have grown to billions of chemicals, virtual chemicals that are synthesizable and stable enough to be synthesized and kept and used as drugs. And then when we have billions of these chemicals available in these libraries, then it is difficult to test them and see the docking, whether they fit into certain proteins or not, because it requires a lot of computational work. That is why artificial intelligence can be helpful. And I have examples of these later on as well. And I'm conscious that I don't have a lot of time, so I'm going to pass some of these. So we have databases, a lot of these databases available. So the amount of number of virtual chemicals available in 2004 was below 1 million, but we now have 37 billion virtual chemicals available. In terms of the protein data, we have 200K unique proteins available in protein data bank. So these are not virtual, these are real three-dimensional structures. We have bioactivity data. So an example is Campbell database. We have binding of 2.5 million chemicals against various protein types. And then we have gene expression data, meaning that we have the effect of drugs on gene expression when they downregulate or upregulate the genes. So we have these measures on other databases as well. And we have, again, gene expression, but toxicogenomic data on live animals. This is not a huge database, obviously, because it is working using a live animal. So the chemicals are injected and then samples taken from tissues, and then we look at the gene expression. And then we have databases that give us the chemical protein interaction network. And one of the widely used ones is called STITCH database, which gives us, so for example, we have aspirin, which proteins it binds, and then there's the relation between the network of the protein that are working together. In addition to that, we have longitudinal cohort databases of people. So for example, UK Biobank has more than close to 1 million actually participants. And this is the data gathered since 1980s. So they have followed people over the years and gathered all sorts of information from X-ray or MRI, MRI data, imaging data, to their lifestyle, to the drugs they're taking, the disease, the race, the genetics, and everything. And then these data are available for artificial intelligence to be used for prediction of, for example, drug outcomes. Within this UK Biobank, we have 31,000 patients who suffer from depression. This is just an example of a disease. And then for these patients, we can look at the use of antidepressants and see the real patient outcome and predict what works best for different patient groups. We can predict, we can see the impact of aging, we can see the impact of metabolism, diabetes, inflammation, and all of that on the choice of the drug as well. So I found that there were 338 papers published using the UK Biobank data, using data science. There is a US equivalent, a US All of Us database. And this is an example that shows that using UK Biobank data, we could see that people who are suffering from rheumatoid arthritis and take a drug that is for rheumatoid arthritis called methotrexate, they are less likely to get dementia later on in their life. So these are the types of information that can be helpful for patients. So with this increased size of the data, it is now possible to use more complicated machine learning algorithms. So I gave an example of simple regression equation, but we have decision trees, which is also very simple. It's just basically you look at the molecular structures, and then if their molecular size is big or small, you put them in the groups. And maybe if the drugs are small, so the computer software runs analysis and identifies properties in the molecular structure that determines whether they will be potent or weak. For example, the computer software may identify that if the structure has 2 benzene rings with, 1 oxygen atom and so on, it will be potent. So that is an algorithm for us for future predictions. But if we have enough size of data, we can move on to black box models, which contain not just one decision tree, but a lot of decision trees. And then the prediction will be better because we can then get average or consensus prediction for each molecule, for each drug. And then we have neural networks, which are even more complicated because they work in each of these nodes. They have different functions, and each function and signal has their own weights that have to be trained so that they can predict the best. So basically, we train this so that the prediction for the data that we have is the best. And if it is the best, we hope that the future predictions for the future chemicals will also work. I'm going to skip explaining this. So there is a difference between current QSAR and the older QSAR methods. The purpose of this slide here is that all computational modeling is not just artificial intelligence. There are molecular modeling involved, homology modeling, protein modeling, and design elements. So we measure interaction energies and design better chemicals using structure-based drug design as well. But artificial intelligence is helpful because it is difficult and computer intensive to simulate the molecular and identify molecular structures and simulate and identify interactions and the surface properties and other properties of these molecules. When you see in here, we have a protein molecule and a lot of water molecules around it. And if we want to simulate individual molecules, the movements and the kinetics and the energy of these water molecules and their impact on the three-dimensional folding of the protein and different temperatures impact on these proteins, it's going to be hugely computer intensive. What they do these days is that they use artificial intelligence to predict what the impact of each of these are going to be, because artificial intelligence is less computationally intensive. So they combine simulation with artificial intelligence. This is an example that shows that even for docking, we can employ artificial intelligence to predict what the docking results will be for future chemicals. As I said, we have billions of chemicals that we can do docking, and we require to obviously a lot of computer time is required, which is not possible. So therefore, we can use artificial intelligence to predict the result of these sorts of simulation. A major breakthrough was AlphaFold, which is an artificial intelligence system that predicts the protein structure. And based on this AlphaFold alone, we can predict structures, the three-dimensional folding of new proteins. And then for these proteins, we can design new drugs. And in fact, there are examples of new drugs that are in clinical trials these days that have used the likes of AlphaFold, which was discovered in 2022, I believe. Examples of companies, so all drug companies are using these artificial intelligence systems because they get the data, they synthesize a new molecule, they then test them, then they use the new data in their artificial intelligence system to improve their artificial intelligence system and come up with better predictions, and then those predicted chemicals are synthesized again and tested. So and this cycle continues. So artificial intelligence helps quicker, faster, and cheaper drug discovery. There are some examples of AI-heavy industry. Exscientia in silico medicine and Benevolent AI are examples of the pharmaceutical industry, which started based on artificial intelligence and not just artificial intelligence, by the way, computer-based drug discovery, which includes artificial intelligence. And these are examples of Benevolent AI, which is one of these companies, drug pipeline, discovered basically initially based on artificial intelligence prediction. Another example of, from the same Benevolent AI company, is a system that retrieves from scientific literature, retrieves data and information based on the weight of evidence. So when you search for information, you say, what is the promising drug? So mask could be filled with a set of potential drug targets. What is the target what is the promising target for rheumatoid arthritis. And then it looks at the literature, searches the literature, and looks at the weight of evidence, not just the best prediction, but the one which is most interpretable, and identifies that, for example, this one, which has lower prediction ability, but it has best evidence based on explanation. So it can identify these, and it is a language model similar to ChatGPT. It's not generative, but it is a language model that that does text mining and identifies the best evidence from the literature, because otherwise we can find multiple answers to the same question otherwise. This is just examples of a list of drugs based on which have been discovered based on AI and are currently under clinical trials. Okay, so I'm going to move on to managing expectations. It is not all rosy, basically, in drug discovery. Unlike artificial intelligence, we don't have as much data. In artificial intelligence, in a chat GPT and text mining, we have a lot of human text available in the internet that empowers ChatGPT. We don't have that amount of data for drugs, even though we have a lot, but we don't have that amount. So it's going to be challenging. Also, we have a complexity of systems that require complex data collection strategies. There is a lack of all-encompassing, standardized, labeled benchmark data sets applicable to a wide range of diseases. So we don't have a set of data that can be used for all diseases that is that could take advantage of artificial intelligence. Otherwise, local systems can work better because the power of artificial intelligence is when it can analyze multiple sets of data together. High throughput data collection is focused on specific research questions. Therefore, there are confirmation bias within the data available. And also phenotypic patient data is lacking. A lot of data is hidden in the text and not available for research. An example that I have here is from my own research for the prediction of the drug side effects. We downloaded data from a side effect database, and then for these drugs, we identified protein binding to different proteins, to more than 6,000 proteins. And then, as you see, some of them give us a good, some of them are good binders, some of them are excellent binders, and so on. And you see the number of chemicals that are good binders to individual proteins, and weak binders and non-binders. You have to, I should mention that these targets are not all the real targets, not the intended targets. So a drug enters the body and it can bind to multiple proteins. Some of these proteins might be intended targets, some of them are not. And you see within this, we have 10 million missing data. So for 6,000 proteins and 1,500 drugs, we have 10 million missing data. So the data availability is not huge. And we have to make sure that we never have a perfect output. We need to understand that the validity of these models have to be tested. Producing a model is easy, but whether the model works or gives us hallucination, like hallucination that we see in ChatGPT, giving us wrong answers, is possible. So therefore, we have to check that these models are valid so that they can be useful in research. They have to be interpretable. Interpretability is a good thing, but we don't have interpretability if we use complicated systems like these black box models, the complicated neural networks. Therefore, it is difficult, and generalizability of these, because they are not applicable they might not be applicable to future AI. I'm sorry, I have passed my time, but I will display the conclusion. And I think I have mentioned whatever conclusion that I wanted to mention before. So I'm going to stop my presentation here.
Thank you, Doctor. And not a worry about time at all, of course. That was absolutely fascinating. So I'm sure our guests don't mind a couple minutes over. It's not too bad. But I also want to make sure that I'm cognizant of your guys' time. I know how busy you guys are as being researchers and faculty members. I will probably have time for like one question. If anybody has it, please feel free to type it in the Q&A session. But while we are, kind of waiting to see if one of those questions does come in, I do have a question, and I always ask our faculty members this: why NSU? It's one of my favorite questions to ask because the answer is always different.
Sorry, say it again.
Yeah, why did you decide to come work at NSU at Nova Southeastern?
Oh, okay. Sorry, family reasons. Coming to Florida is always good, sunshine. Coming from London, always raining. But yes, and I like Nova Southeastern University as well. So I had applied to come here before COVID, but due to COVID, I didn't come.
Fair enough. Fair enough. We do have I do have one question. It was kind of going back more, the topics are more back in the beginning of your lecture, but it was in drug discovery, what problems can happen from like a modeling standpoint when using AI? Because as a lot of people know, AI is sometimes not the best, depending on what you're using.
Yes. So problems that can happen is that you always can get false positive or false negatives. So false positives means that you predict that a drug is going to be effective, but then you synthesize it, and so obviously a lot of investment, and then you test it and you find that no, it doesn't work. Sometimes this is an easier problem, but the more difficult problem is false negative, when your model can miss chemicals that are potentially very, very useful, but model predicts that they are not going to work, so you don't test them and you miss.
Awesome. All right, Doctor. Well, I think that will wrap us up this evening since like I said, know we are a couple minutes over. But for those of you that are watching, have been watching tonight, thank you very much for being here. For those of you that are watching later on our YouTube channel, feel free to contact us if you have any further questions. And please, everyone, keep an eye out for our virtual open house that we'll be having next month. So you'll get some emails and communications about that down the road as we get started with all of that. So we're very excited for that virtual event as well. With that being said, Doctor, thank you so much for taking the time this evening. Members of my staff, thank you for being here. Dr. Reza, always a pleasure to have the department chair with us tonight. I hope everyone has a great rest of your week and look forward to seeing you guys all around campus. Hope everyone has a great evening. Take care.
Thank you.